Welcome to Faculty Focus, the podcast of the Faculty of Pharmaceutical, Biomedical and Veterinary Sciences of the University of Antwerp. I am Thanopoulou. And of course, as always, Marleen is joining us today as well. Good morning, Marleen.
Good morning.
In research talks, we meet with researchers from our faculty and discuss their latest work. And for this episode of Faculty Focus, we are going to talk about the OncoProTools Doctoral Network, a consortium funded by the European Union that wants to force breakthroughs in cancer diagnosis and therapy. And for this, we have Professor Pieter van der Veken with us, and Pieter is one of the principal investigators of the Medicinal Chemistry Group of the University of Antwerp and coordinator of OncoProTools. Good morning, Pieter.
Hi, everyone.
And we also have Nikita, Shikut and Magda Thanopoulou with us. Both are doctoral researchers in the OncoProTools network. A good morning to both of you as well.
Hello.
Good morning.
So, let's kick off with a general question, Pieter. What exactly is the OncoProTools doctoral network?
Okay, so we're a Marie Curie doctoral network, and we're a consortium that is a group of research groups, both from companies, biotech industries, and academic institutions. And we are coming from 8 different European countries in our case. And we work on a central team, and the team is proteases in the tumor microenvironment. And I understood that we will delve into that topic a bit more later. So I'll stick to that description for now. And we get funding from the European Commission to develop new diagnostics and treatments for cancer. That's in a nutshell what our network is about.
Okay.
And the most important thing, I almost forget, Teun, is that while doing that research, our goal is to train 10 young researchers, PhD candidates, to make them the future leaders of research and development in Europe.
Okay, and we have 2 of them here today.
Exactly.
Great.
So we have Magda sitting next to me and Nikita in front of me.
Great.
Your consortium is very big. You have a lot of stakeholders. What does collaboration within such a consortium look like?
Well, as Pieter introduced, the consortium looks very complex. I will try to make it less complex. And so simply, we have a lot of scientific groups that work on different projects. We are divided by 3 big groups that collaborate, for example, in our case, on some small proteases that we'll discuss later, maybe. And among each other, we coordinate our work. We are very complementary to each other. So simply some expertise that we could provide, we give to our collaborators, and our collaborators are giving those expertise that we lack, for example, here at the University of Antwerp. And then we benefit each other's work and projects. So,
Okay, real collaboration. So
yeah.
Europe has a high cancer burden. In 2020, 2.7 million European Union citizens were diagnosed with the disease and 1.3 million lost their lives to cancer. This toll is expected to increase even further, making cancer the leading cause of death in the European Union by 2035. So the European Commission called for a big push in cancer research, and they published its Europe's Beating Cancer plan. And Pieter, how does OncoProTools contribute to this ambitious plan?
Right. That's a very difficult question, Teun. So I'll try to keep it simple. The European Commission's plan, so Europe's Beating Cancer plan, is actually a very all-encompassing cancer management plan that spans things like patients' quality of life, novel diagnostics, novel treatments, but also very broad cancer prevention related topics. So alcohol policies and smoking policies are also part of the Europe's Beating Cancer plan. And of course, we can only contribute to a very small part of that. And I think I'm not lying when I say that we mainly contribute to diagnosis and treatment of cancer.
Okay.
So development of new diagnostics, development of new treatments, and especially treatments and diagnostics that are patient-friendly, so that respect the patient's quality of life as much as possible. So we develop non-invasive treatments and diagnostics, for example, PET scanning-based techniques, treatments that you can just inject into a patient and that autonomously find their way to the tumor.
Yeah.
So without affecting the healthy parts of the patients, these are the things that we typically do.
Yeah.
You say, Pieter, it's a small contribution, but especially in such a big plan, I think every small contribution is equally important, no?
Yes, of course. So I think there is a lot of things that still need to be done in cancer diagnostics and treatment, and I'm happy to contribute to it.
So it's the sum of all those small research that contributes to a push back in cancer.
That should bring us to the next level. That is true.
Yeah. And so why is it important that this research is being conducted by young PhD researchers at the beginning of their scientific career, Magda?
So it's to be trained to become good researchers in the future, and we aim to also have a good skill set, very interdisciplinary, as we already mentioned, and also socially engage as much as possible, be able to explain research to people outside the field, and generally spread the word out there.
And you say the word interdisciplinary, but you have two more I's. You have the intersectorality and the internationality. So what does it mean, or in the context of this project, at least?
Well, maybe I can elaborate a bit on that. So simply, those are the categories from Horizon Europe program. So, and meaning that, of course, the PhD students, when they study, they have huge expertise on the hard skills, on the science that they're doing, but also the European Commission puts efforts to develop different flexible mindset of the PhD researchers, meaning that we could change between disciplines, meaning that we could transfer our skills from academia to industry and vice versa. And then, for example, the second I was interdisciplinary. Well, here it's actually very easy because in such a huge collaboration, we have a lot of different fields presented. For example, medicinal chemistry in our case. Then we also collaborate with Laboratory of Medical Biochemistry, which is enzyme kinetics, and also with USA, with MICA groups at the University of Antwerp. Well, of course, another groups within our consortium, so which focus on animal biology, on tumor biology, on different skills. So that's for interdisciplinary. So we migrate among each other, learn new skills, and
infect each other.
Well, I wouldn't call it like this. Well,
with theory and knowledge, right?
Exactly.
And competences.
Well, in positive meaning, of course. And internationality, well, of course, I mean, I'm personally Russian and Magda is Greek, and we change our cultural backgrounds, and we develop our career paths in different cultures and in different countries. Also, we have so-called secondments. It's short stays in different companies in different countries as well.
Okay.
So, for example, for Magda it was in Germany. For me, it's Switzerland. So we had to go there.
And that's a part of your PhD that you have to...
Yes, exactly. That's a part of the OncoProTools studying.
But it's also nice to mention here that generally, basically, everybody of us is from abroad, living in a new country. So it's very, very international. Yeah. So that's very interesting. Also, you get to meet new cultures. You have to collaborate with them. So it's nice to learn these soft skills.
Yeah, it sounds very inspiring indeed. Yeah. But collaborations across disciplines, sectors and countries, it's not always straightforward. Were there moments when things became difficult, and did you feel that you had the right support to navigate those challenges?
You always encounter difficulties, but of course you can always ask around supervisors. Any supervisors, of course.
Magda is pointing to Pieter.
And then, of course, because we are so many people in that consortium and so many other PhD researchers, you're not alone. You can always ask them. They're going through the same thing pretty much. But generally, the good thing is that we have flexibility to do what we feel is right. Of course, we have pointers by supervisors, but it's very nice to be flexible about things like the secondments that Nikita mentioned.
I can actually give an example about these three secondments that I already introduced. Well, I actually didn't do it because there were some bureaucratic issues, and also some of the experiments that we would do there were not really applicable anymore to the projects, and we could replace them with some expertise that already we have at USA. And I was like, well. And then eventually, so the European Commission and also the supervisory board, they were very open to my suggestion to shift it.
So you're very much in charge of your own trajectory.
Yeah.
So as long as it makes sense, for the project, for the personal development, so it's fine. Yeah.
Okay.
It's also, I think, nice to mention that the secondments generally apply to one other academic environment, like one other university, and also in the industry. So you can see also both perspectives.
It's also a mix there.
Yeah, you can have a mix of different work environments. So that's also nice.
Yeah.
And many universities have ombudspersons or confidential counselors in place. Do you think these support channels are visible and accessible enough for young researchers? And how important is it to have these support channels in a large project?
Well, we definitely saw some emails from Pieter. University of Antwerp has a very good information.
I'm an ombudsperson.
I was about
PhD researchers.
I was about to say it.
Yes,
actually,
thank you.
I saw
You saw my mail?
Very good, you exist.
yes, I saw your mail. That whenever
Okay,
we
I'm
have
happy now.
issues, well, but as far as I understand the concept of ombudsperson, well, it's when you have a conflict between different parties, there would be a neutral person that would mediate it. And,
already in procedures,
Yeah, with the procedures, but so far, well, we didn't have any critical situation that would require a ombudsperson, but
thankfully.
Thankfully, yes. But answering your question, well, it's visible in the University of Antwerp. Also, we have our own ombudsperson. That's the OncoProTools. We never used the expertise so far because we
thankfully never needed to.
Let's say formally we have it. So far we didn't need it. So
no, but you know that it exists and you can contact
exactly.
someone. That's important, right?
Yeah, exactly.
Okay, maybe let's talk research. So the project is shifting focus towards small molecules instead of the larger antibodies that have traditionally been used in cancer targeting. Why is that?
So small molecules are small. So that means they can penetrate tissues much more easily than bigger molecules, due to size, and also they can do it that faster. So yeah, that means that they can target the cancer better. And also talking about larger scale synthesis, it also is easier to access small molecules like synthetically to make them. So that means that the treatment is more accessible and generally cheaper.
Okay.
So.
And what was the problem with the larger antibodies that they normally use?
So they cannot really penetrate that much the tissue.
Okay.
So they're not as
so
efficient as effective.
And can you explain the importance of a vector unit in the context of tumor targeting strategies?
So the vector unit is what recognizes the tumor. So it's that what makes it selective towards that tumor and not the other healthy cells of the body. So this is what generally standard and cancer treatments do. They just target every cell in the body, killing everything, including the tumor, which is good. But the bad thing is that you also target the healthy cells. So that's very heavy also for the patients. So by only affecting the tumor, then it's better for the patient. It's more patient-friendly.
Yeah, that's what Pieter referred to with the less invasive methods.
Yeah. And the importance of the tumor microenvironment here?
Maybe I can elaborate on that. So the tumor microenvironment is unique space actually in the body. First of all, its main function to promote the growth of tumors. So it promotes the tumors to grow, which is a negative thing, of course. But also, it's a very important and very interesting target for tumor oncology, for the oncology itself, for therapeutics. So we could target some specific cell. We would disable a bit the tumor microenvironment cells, and then it would decelerate.
So it's like a barrier or something around the tumor.
Yes. Yeah, so that keeps the tumor growing. And then also in the tumor microenvironment, there are a lot of small targets that we could specifically, I would say signatures that for each tumor microenvironment. For each tumor, they are very unique, and so we could put, for example, breast cancer, prostate cancer. They have their own tumor microenvironments, which are different from each other, and then we could explore them, and we could select some targets. We could create some specific knowledge about it. We could target it. We could specifically treat it, and so it's very important.
And for these proteases are very important. Can you elaborate on that a little bit?
Proteases is just a very, it's a type of protein that's, by definition, it's a type of protein that cleaves the peptide bonds. In terms of tumor microenvironment, they just eat big proteins, make it smaller, and then more digestible for other bodies that's consuming the rest of the biological material. Well, for us, it's kind of a Pac-Man. It's a protein.
The yellow man.
Yes, but the good thing about this Pac-Man, that's if we, for example, put some target molecule to this mouth, then we could see. So we could visualize this Pac-Man within the tumor microenvironments. We could treat the Pac-Man, and we could do a lot of things with some specific tumors. We could treat them, we could visualize them.
So it make them good candidates for tumor targeting.
Yeah, exactly. Yeah.
And the benefits of this approach, why is it so important?
Well, I briefly mentioned that every tumor and tumor microenvironment is unique. So then simply the benefits is, well, it's a called selectivity, that we could distinguish one tumor from another. And for example, if we know from biopsies or from different assessments that this patient has some specific type of tumor of the cancer, we could select the treatments, we could select the visualizing agents, we could select the therapeutic agents, and we could treat it more specifically and more precisely. And yeah.
So personalized treatment.
Yeah.
Okay.
So very promising.
Yeah. So you both have been working on this PhD for 4 years now. Can you share a memorable moment from the past 4 years?
Yeah, a lot of them.
Any, Magda?
I have a lot. But I think maybe the first summer school and annual meeting is one very much worth mentioning. So it was the first time you meet all the other collaborators in person. In that setting of like 15 days, it was like a long time. Maybe even less, maybe I'm exaggerating. Maybe like 10 days. But we have this summer school when there are different people giving talks in different topics. We focus both on soft and hard skills. And we also basically, it's like school, but we also contribute to conversations. So it's not like passively listening to information, it's also contributing and thinking collectively and doing small projects. And then the annual meeting where you had to present what you had done so far to all the supervisors in the consortium. So of course the first time, it's a bit stressful. But everybody's presenting, and then you get good feedback, maybe potential new collaborations depending on your project. So it's very interesting and very nice, and totally memorable.
Yeah, I would agree with Magda as well. But to my mind, actually, it came like. Well, a conference came that we visited this January. So we went all together to South Africa, to Cape Town, and it was a huge diagnostic conference. And then, well, for me, what was interesting that you could, well, first of all, in live see these people who are passionate about the research, also, well, listen to the talks, and also afterwards communicate with them. And it's a first experience like this for me. And then, of course, South Africa itself. I've never been to Africa before, and it's
a highlight.
That would be a highlight for me.
And do you feel that you've made an impact in the field of cancer research?
I mean, how would you define impact? I would say
small steps.
Small steps, for sure, yes. Because, for example,
all important.
Yeah, for sure. I would say that, for example, in my project, we started from synthesis of different inhibitors. So we work with one of the proteases. It's called granzyme B, and then. We developed a lot of compounds that would target this small enzyme, and then eventually we came up with a probe that we are currently testing in mice models, and I would say it's small impact for sure.
And another goal is to deliver 10 top researchers, that's the goal of the program, not only as excellent scientists, but also as socially engaged researchers. But what does this all mean, and do you feel like a top researcher? It would give me stress.
Yeah, of course, I agree with you. It's a label, of course, but well, let's put it as a researcher, yes, and good quality researcher, and I would say yes. But answering your question, socially engaged, well, it's another goal of actually Horizon Europe programs that we would be able to communicate our science to broader public. Here, I would mention that we have a partner on board. It's called Cancer Patient Europe. It's an organization under the European Commission umbrella. They advocate for cancer patients, and they organize a lot of gatherings with different experts. Sometimes we were invited as well, and we could also share our opinion as a cancer researchers. And for example, sometimes about our regions, there was a round table about different regional policies on cancer treatment and diagnostics. So I was somehow advocating for Eastern Europe, and
it's nice to see the other side of it, because we're in the lab, we're in the very early stage of that research, and then you see the other side of the patients. So then you know you can have an impact on them, and that it's good. And generally, we try to communicate a lot about what we do. Like we have a lot of communication campaigns with social media. Like you can find us on Instagram, on LinkedIn, on YouTube.
And now in a podcast.
And now this podcast, of course. So yeah, that's also nice.
Yeah.
And yeah, of course, the hard skills with different disciplines involved and learning different things and collaborating with everybody.
And Pieter, do you see a difference in the new generation of scientists?
Well, I think too, that focusing on soft skills, for example, has always been part of Marie Curie actions from the beginning onwards, and already when I was a student that existed, but it was not formally implemented by doctoral schools at normal European universities, and I think that the Marie Curie actions have really helped at bringing that soft skill training to all PhD students, in fact.
Yeah. So it's more institutionalized than it was
It's
before.
more institutionalized for sure.
Yeah.
But I do think that even now Marie Curie students have a more formalized training package with that respect. Non-Marie Curie PhD students, they can choose their soft skills trainings themselves, and I think that is not always thought through to the same extent, I think, as what Marie Curie networks do for their students.
Okay.
So the quality of your soft skill training is also evaluated by the evaluation panels, and if they don't like it, if they think it doesn't make sense to focus on this or that topic, then your project will not be approved.
Okay, so it's really important. Yeah. Okay. And so maybe to conclude, Pieter, what does the future look like for OncoProTools?
Well, there's different types of futures. So most of all, the
positive one.
Future. So with Magda and Nikita's PhD projects coming to an end at the end of this year, we will submit a follow-up project for OncoProTools.
Okay.
But you have to keep in mind that approval rates are below 10% these days. Yeah. So chances are that it will not be approved immediately, and that we will have to resubmit it maybe several times before we will be successful again.
Okay, let's hope for success.
Cross my fingers. Yes.
And
Nikita and Magda, what does your future look like?
Well, I would say for me, I would just focus on some everyday things. For example, finishing my thesis, defending it, and I would further move to an industry and try myself to get more experience there and see how it works for me, and then shape my career path coordinately. And
okay.
We'll see. I don't know what the future brings.
Yeah, for me also, first goal is to defend the thesis and become a doctor, of course. And then secondly, probably also join the industry, but I don't really mind staying in Belgium or moving again, so it's very open for me. So depends on job positions that are open.
Okay.
So
all right.
We'll see what the future brings.
That's a good, also cross our fingers.
Thank you.
That's a good conclusion for this podcast. Thank you very much for coming by. Good luck with your career, and Pieter, good luck with the new application.
Thank you very much.
Thank you for inviting us.
Thank you.